Okay, good evening. Thanks everyone for joining tonight in the middle of summer on July 24th. We're really excited to have Zoe Stacey presenting on whirling disease and what it means or could mean to us here in British Columbia. 2024 has been a big year in the world of invasives and disease. We've got CWD now. Mussels are getting closer, it seems like, every year. And we have some more, a watershed that has whirling disease now. So tough year for the land of invasives and kind of invasive diseases. So we're bringing you some experts to talk to us and let you ask your questions regarding whirling disease tonight. Format for tonight is very similar to all of our other conservation webinars. It's going to be presentation will be 40 to 45 minutes. We'll do some Q&A. You can put your questions into the Q&A box, and we will aggregate them at the end, and then we will ask Zoe the questions as they an expert. She is not a policy maker or a decision maker. So keep your questions, I guess, more in line of the world of technical stuff. She also works for the Government of Canada, so we have to ensure that we are asking questions that are consistent with her role with the government. Quick little bit about Zoe. She works as an aquatic animal health veterinarian for the Canadian Food Inspection Agency. You all would have missed her background, but she's got a ton of work. Started on the East Coast and has worked her way slowly west, but a lot of work on disease outbreaks and those sorts of things. So she works on whirling disease and works with a number of freshwater, the hatcheries across the province as well. So with that, Zoe, really appreciate you coming on tonight and engaging with all the folks who are attending the webinar. And if you want to share your screen, we'll turn it over to you.
Thank you very much for the invitation, and I'm very pleased to see the people that have joined. I'm looking forward to sharing some information on whirling disease and answering any questions you may have. Okay, can everybody see me?
Looks good.
Okay, so today I'd like to speak with you about whirling disease in Western Canada. And at the end of this presentation, what I'd like you to understand is the whirling disease background and history in Canada, what our CFI area declarations, that is the declarations of infected areas, mean, the current whirling disease declarations for Canada, and the impacts of declared area status. I'd also like to discuss the current situation in the Columbia River watershed and future plans. So the whirling disease pathogen the disease is caused by a parasite called Myxobolus cerebralis, which is a cnidarian, which is interestingly in the same phylum as jellyfish and anemones. It has a very complex life cycle that includes an annelid worm, tubifex, as an intermediate host. The infection has only been reported in salmonid fish and specifically freshwater salmon and trout. And it is only transmitted by horizontal means, which means that it can't be transmitted from a parent fish to a fry, but only between fish. The organism has two spore forms, which can make it very difficult to eradicate. The worms release a free-floating actinospore, which we also call a triactinomyxon, or TAM. That's the infective stage for susceptible species of finfish. The actinospore is attracted to the external mucus of any species of finfish, but it only tends to cause disease in those salmonids. The TAMs migrate through the skin and the other tissues of the fish until they reach the cartilaginous tissue, generally in the head and spine, where they settle and eventually form myxospores. It's most commonly found in the cartilage of the head and of the caudal peduncle, which is the end of the tail just before it turns into the fin. Myxospores are released into the environment when infected fish die and decompose, or through feces of flesh-eating predators, mainly piscivorous finfish, so predatory salmon, for example. It can be found in the intestines of some birds as well, but the link between that and infection is not as clear as through the fish. The myxospores settle into the sediment where they are eaten by the tubifex worm. Here's a diagram of the life cycle. So we'll start with the intake of spores by the tubifex worm. It becomes infected, and then in the intestinal epithelium of the worm itself, those actinospores develop. And then that as the worm either dies or it defecates, those triactinomyxons are released, and they can then infect the trout through the skin. After migration, the TAM goes into the head and the tail of the fish, which is why you see this darkened tail. The fish, if it doesn't die as a young fish, if it gets to an adult size, these spores will transform into the myxospore, which is the difficult spore to eradicate because it's quite tough. And then that will be released when the fish decomposes so that the life cycle can begin all over again. So what do we see in these fish that are infected by Whirling disease, by Myxobolus cerebralis? So in the population, mortality is highly variable. It can be as high as 90% in some situations, especially in younger fish. So we tend to see the disease, the clinical signs, and the mortality in young of the year fish. However, we have not seen that in either Alberta or British Columbia. We have detected the pathogen, but we have not seen high mortality in any populations of fish. In terms of morbidity, which is mainly behavioral, we see a decrease in feed conversion ratio when animals are infected in a hatchery situation, and the abnormal swimming, which is what gives the disease its name. As clinical signs in the fish, you see that caudal peduncle just before the tail fin can become dark or even black. You see spinal deformities, mainly scoliosis, so the spine gets kinky. There are deformities of the cranium, so you'll see sometimes that the mandible, the lower jaw of the fish, becomes shortened, so they look like they have an overbite, or they have a depression or a dent in the top of their head. And sometimes you can see shortening of the operculum, so the gills are exposed, and sometimes opercular cysts. On necropsy, the cartilaginous tissue is disintegrated. It's not necessarily visible with the naked eye, but when looking at things under a microscope, you can sometimes see that disintegration of cartilaginous tissue. And you'll see those skull, vertebral, and opercular deformities that can sometimes be seen on physical examination. So here we have an example of clinical signs. These are young salmonid fish. So you can see here the darkening tail in this fish, and there's a bit of a kink in it. This one has the dark tail but no kink. And then here are some hatchery raised fish that were infected that it's really pronounced. You see a reduction, well, this guy's pretty fat still, but you can see a reduction in feed intake and some severe deformity of the tail. And this is what it looks like in a tank. So you can see the fish is spinning around, which is where the whirling comes from, and it's got that really distinctive black tail. So if you see a fish like this in the water, it's not a definitive sign of Whirling disease. It's possible that there could be other things that cause it, but it is definitely a concern. And that's the sort of thing that we look for in some of these populations. However, I should note that in the ones that we've seen that we had the detections.
We're going to make a spicy and numbing fish from the properties of Sichuan.
Sorry about that. YouTube. In some of these, the fish that we have detected the pathogen, we have not necessarily seen clinical signs. So it can be there even if it's not obvious. So when we're considering the fish to sample, I mentioned before that it tends to be found in young of the year fish. So we want to look at species susceptibility. So looking at freshwater salmonids, salmon and trout. We look at the population size to determine the sample size, and most important is the age of the fish at the time of testing. So The little fish on the left is much more of a target than the giant fish on the right. So what about whirling disease? Well, it was first detected and described in Germany in 1893, and it was present in European brown trout. However, they showed no clinical signs because they had evolved with the pathogen, and it didn't cause outbreaks or signs of disease. So the detection was actually in rainbow trout that were imported from North America and stocked into rivers and lakes in Germany. It has since been found in North and South America, Africa, and in New Zealand. In Canada, the first detection was in a wild salmonid, likely a brook trout, in Johnson Lake, Banff National Park in August 2016. It was subsequently found in various locations downstream on the Bow River and its tributaries. The first detection in an aquaculture facility adjacent to the Bow River occurred in 2016 also, and that farm moved fish to other aquaculture facilities and to wild water bodies in the province for stocking purposes. And that was a large contributor of the spread of the pathogen within Alberta. Tracing of fish movements from this original infected premise found 7 more infected farms, which depending on the farm, they were either able to cull the fish and eradicate the pathogen, or they remained under quarantine in some cases until we determined the disease status of the entire watershed. It did not cause severe disease in any of these places. So it was another instance where it was detected, but we didn't see mortality and we didn't see any severe disease. As a result of the stocking, many lakes and dugouts were also contaminated. So in the end, after several years of surveillance by CFIA, Parks Canada, and with the government of Alberta, it was found in 4 major watersheds. So most of the central part of Alberta is considered infected with whirling disease, starting with the North Saskatchewan River in the north, then the Red Deer and Bow, and the Old Man in the south. Once a pathogen is found in wild fish, eradication from a watershed is not possible. We just can't catch all the fish that might be infected, and in this case, also the worms. So in that case, we move from a disease response situation to a risk mitigation approach. And that's where domestic movement controls and surveillance in adjacent watersheds come in. CFIA did some surveillance with the government of British Columbia in the Columbia River in 2017 and 2018. We collected and tested about 575 samples, and we did not detect the pathogen. Yoho National Park staff continued to do some surveillance in their park waterways due to their proximity to the index site in Banff and the amount of traffic that moves back and forth. The samples were submitted by Parks Canada to a private lab in BC in September 2023, and that resulted in a suspect case. However, we couldn't confirm the pathogen, so there are several steps that our labs take to confirm that the disease is actually there. So there was quite a bit of additional sampling that had to be done by Parks Canada in and about Yoho National Park. And finally, we did detect it and confirm it in Wapta Lake in December of 2023. Well, I have some information here. I'll show you these web pages at the end of the presentation. But what I've shown, what I've put here is an information page on detections and zoning of history of whirling disease, and a whirling disease fact sheet that you can access. I will be sending an email to the organizers after this webinar that will include this information to be sent out or posted so that you all have access to these links. So now that we've found whirling disease in British Columbia, what are we going to do about it? Well, that's where the National Aquatic Animal Health Program's Domestic Movement Control Program comes in. And there are some definitions I want to go over so you understand the zoning decisions that we've made. An infected area is where the reportable aquatic animal disease is known to occur in Canada. And there are several of those that you can have a look at some disease tables on our website with those links that you'll receive later. A buffer area is where the reportable aquatic animal disease may occur because of a link to an infected area, but it has not been detected in that area. And a free area is where the reportable aquatic animal disease does not occur. Again, here are some pages I'll show you at the end, but there's some general information about the Domestic Movement Control Program and the disease declarations by province and watershed. And this is what it looks like for whirling disease. So as you can see, Alberta here on the bottom, those watersheds I mentioned are all considered to be an infected area with whirling disease. And because there are movements between of fish and people, water, equipment between the infected areas and some of the other areas in Alberta, we consider those to be buffer areas. And that's a bit of insurance to make sure that we can control the movements from those buffer areas into other parts of Canada that are considered free of the disease. So that we can do our best to contain the pathogen and prevent its spread to other areas. Here in British Columbia, we have a relatively complex system in that part of our province drains into the Arctic Ocean watershed and part into the Pacific Ocean watershed. And then we have the declaration for the Columbia River watershed as an infected area with whirling disease due to that detection and confirmation in Wapta Lake. As with Alberta, other watersheds in British Columbia are considered buffer areas for the disease because there are epidemiological links between the two, the multiple watersheds. So, and that allows us to control movements, but it also gives us some focus on where to do additional surveillance to try and determine whether this pathogen has spread any further than in the Columbia River watershed. This is what our watershed map looks like. So this is Alberta here. Here's the border of the Columbia River watershed, which pretty much mirrors the Rocky Mountains. These are the watersheds in Alberta, 5D and E, C, B, and A, that have all been declared infected with whirling disease. And this here, the big one, is Columbia River watershed, 08N. So all of the rest of the province, whether it's in the Pacific Ocean watershed or the Arctic Ocean watershed, is considered to be a buffer area. And we are working to do some surveillance in the watersheds adjacent to 8N to try and figure out whether the pathogen has spread any further. So what do these area declarations mean to stakeholders within them? Well, CFIA disease responses are based on whether the detection occurs in an enzootic area or endemic area, which is infected, or a non-enzootic, which is a free or a buffer area. So if we found whirling disease within the Columbia River watershed, again, we would not go into full disease response mode. However, if we found it outside of the whirling disease infected area, then we would institute a disease response, and how far we would go with that would depend on the situation, whether it was found in wild or farmed fish. When a watershed is declared infected, movements of susceptible species are subject to the Domestic Movement Control Program. So our domestic movement permits are required if a susceptible species or certain things are moved from an area of a lower health status to an area of higher health status for an end use which is controlled. And I'll go over that at the end of the presentation when I show you the websites. These are the species that we consider susceptible to whirling disease, which, as you can see, encompasses the majority of salmonid fish found in Canada. With a couple of exceptions, lake whitefish are not on there. That's something that there are a few species that we're evaluating, but these are the ones that are of greatest concern and the ones that we want to make sure that they remain healthy populations in other parts of the province. For a movement to be allowed, the animals have to be free of disease, and certain permit conditions have to be met to ensure that the animals are being moved in a biosecure manner. So whatever is being done with these animals, they're being moved from a place to another place where they're not going to spread that disease. So additional considerations, our buffer areas are a priority for surveillance. And there are situations where provinces and other regulators may have stricter controls within and between declared areas. So in this case, CFIA has declared the Columbia River watershed as infected, But the province may, and indeed Parks Canada has put in some controls on movements within that watershed based on their legislation. And that same thing may be done by the province. That has not been the case thus far. The only closures have occurred within the national parks. But it is a possibility that the province may decide to do some additional control measures on the waterways under their jurisdiction. The other thing that we consider is development of educational material to prevent spread and assist with passive surveillance. That's essential to our goals here. We don't have eyes everywhere, and we rely on members of the public to let us know if there's anything that they're seeing that they think is concerning. That doesn't always result in an investigation, but it is nice to have multiple sets of eyes on these waterways so that we have a better idea of what's going on with the salmon populations there. With whirling disease, we have a distinct advantage in that the existing invasive species prevention programs also work against whirling disease. So those things that you're doing to prevent movement of mussels and some invasive plants is also effective in preventing the spread of whirling disease. So our whirling disease zones, as I said before, the Columbia watershed is infected. The rest of BC is a buffer. And we did that for a couple of reasons. For one thing, there are connections between the Columbia and these other watersheds, but the disease has also been found in Alaska and in states to the south of BC, where there are also several connections with Canadian watersheds. So we don't have any definitive evidence that the pathogen is outside of the Columbia River watershed, but it is a possibility. So that's the reason the buffer areas have been instituted. So what's going on? Current situation in the Columbia River watershed. So to begin with, a little bit about the aquaculture. We're lucky in that there are not a lot of commercial aquaculture facilities in the area. It's mainly the Freshwater Fisheries Society of BC hatchery they are moving fish outside of the Columbia River watershed. I should note that CFIA is not controlling movements of fish within the Columbia River watershed, only from within it to other areas outside of the Columbia. So Freshwater Fisheries Society has the Summerland hatchery in the Okanagan and the Kootenay hatchery in around Cranbrook area, and they stock fish in other watersheds. So they have been affected by the change, and CFIA did testing and permitting of their fish to allow them to continue with their stocking activities. You'll be pleased to know that there were no detections. Other farms in the infected zone do not have a history of fish movements outside the watershed. There are certainly movements within it, but that is no longer a concern for CFIA. We are now focusing on preventing spread from the Columbia River watershed. For permitting situations, each facility is evaluated for their ability to meet the domestic movement permit conditions. If they meet the requirements on paper, they are inspected by a CFIA veterinarian, which is me, and samples are taken for Myxobolus cerebralis testing. If the disease is not detected and they meet all of the other permit conditions, then the permit can be issued. And those are issued for a period of time based on testing of specific populations within the hatchery. And now probably what most of you are interested in, what's going on with angling in this situation. I would like to make it very clear that CFIA does not place any controls over the movement of equipment used by anglers. There's no permit required for angling activities in British Columbia, but we recommend that basic decontamination procedures be applied. That is a recommendation, not a requirement, but it certainly will help to prevent the spread. As of May 17th, 2024, it is illegal to transport watercraft with the drain plug in place in British Columbia. And this was put in by the government of BC in response to this whirling disease declaration. So before transporting a boat or other watercraft, owners and operators must remove the drain plug and drain all water on dry land, including all internal compartments such as ballast, bilges, and live wells. The clean, drain, and dry protocol is effective in reducing the spread of whirling disease as well as invasive species. CFIA and the province of British Columbia recommend that equipment be decontaminated after the clean, drain, and dry protocol is complete. This is not a requirement, but it is the best way to prevent spread of the pathogen. An information sheet with disinfection instructions is available on the BC government website, and I'll show you that after the presentation. The use of the basic procedures for preventing spread, clean, drain, and dry, should be applied whenever equipment is transferred between water bodies, even outside of the Columbia River watershed. And that applies to anything that comes in contact with surface water, so a lake or a river, within any water body. Excuse me. In this situation, you may want to consider using equipment that is more easily disinfected. For example, PVC or nylon waders are much easier to disinfect than neoprene and felt-bottomed suits. If you're moving between the Columbia and other watersheds regularly, you may want to consider having 2 sets of gear, one of which is dedicated to use within the Columbia River watershed. We still recommend the clean, drain, and dry and the disinfection, but it can reduce the burden on you and also on your equipment if you don't have to disinfect it as often. From a food safety perspective, fish that are infected with Whirling disease are safe for human consumption. The pathogen can't infect humans, and it's usually found in the head and spine, which is pretty unusual for people to eat. However, fish and whenever you're cleaning a carcass, the carcasses should be disposed of in the garbage. If they're cooked, that's not as much of a concern, but still you want to dispose of it properly. And never throw a carcass or organs back into the water or leave it on the shoreline where it could be scavenged by another animal. In the future, CFIA and the province of British Columbia have entered into a partnership to do surveillance of wild salmonids in buffer areas in BC. And we're at this time, we're focusing on the watersheds that are immediately adjacent to the Columbia River watershed, and we may expand that depending on what we find. Surveillance plans are being drafted. As you can imagine, capturing wild fish for surveillance purposes for taking samples can be quite complicated. So there are a lot of things that we need to work through, but we're working diligently with the province and with Parks Canada on this. We will support additional surveillance within the declared infected area, but we won't be actively involved. We will follow up on reports, but we won't be putting any further control measures within the Columbia River watershed. But we will continue to control movements out of the watershed with the domestic movement program. Surveillance is being considered in Saskatchewan, but will likely be delayed by the detection in British Columbia. Okay, so that concludes the talk. I just want to show you a couple of resources that we have available on the CFIA and the Government of BC website. So this is the CFIA Whirling disease page that includes updates on the zoning for Whirling disease in British Columbia and what's been done in Alberta in the past. Here, there's also a link to locations infected with Whirling disease. So if you click on that link, it shows you when the pathogen was confirmed detected, where, and in which species. As I said, it was found in Wapta Lake in Yoho National Park. That's not something we generally don't post a specific water body, but that information has already been in the news, so I didn't have any concerns about sharing it with you. But as you can see, there are other entries here for detections within Alberta. This is the information page about aquatic animal domestic movements. And there are quite a few resources on here about declarations of disease status, when a movement permit is required. And one thing I'd like to bring your attention to is this right here. So when we're talking about need of permits for finfish, these are the end uses that we're concerned about. These are the ones that we've determined that has the highest risk of transmitting disease from one watershed to another. So most of this is related to culture and also for diagnostic testing and research purposes. So if you want to release a fish if you want to stock a fish, or if you want to move a fish from an infected area to another culture facility for grow out, If you're feeding fish, if you're feeding raw fish to other fish, those are the sort of things that we want to make sure that are being controlled. And then in terms of things, there are a few things on that list, again, mainly to do with culture situations. So things like underwater lights, fish graders, things like that. But when it comes to whirling disease, it also includes the worm that's used. So if an organization wants to move worms around for diagnostic testing, for example, that's when they will need a permit for that purpose. So as you can see, there's nothing to do with angling on this list. And finally, this is the whirling disease page from the government of British Columbia. It has a little blurb here on the pulling the plug. And one of the most useful things here is there's a background and guidance brochure here. And then there's the decontamination protocol. Okay, so this is developed by government of Alberta based on what was done by the government of Alberta. Sorry, government of British Columbia based on what was done by the government of Alberta in response to that detection, the detections there. So there's quite a bit of information on preventing the spread, how to clean, drain, and dry. And then there's specific information on a decontamination protocol, the steps to take, and then the decontaminants that you can use. And there's some detailed information on how to do that in this this guide. I've also been asked by somebody with the government of British Columbia to let you know that if they have developed some signage that can be used to raise awareness around certain water bodies. So if that's something that you want to have installed in an area where you see that there's a lot of fishing, please email this whirling disease gov.bc.ca with any questions that you might have, or if you want to get a copy of that sign that you can put up. And with that, I conclude my talk. I thank you very much for your attention. I'm prepared to take any questions you may have, and I hope you've learned something today.
Thank you very much. We have a bunch of questions lined up here already, and we're straight into the technical stuff. Awesome. So the first one is, it was stated in the presentation that we haven't seen a lot of high mortality, but there is a paper that's was done on the Crow's Nest River, which was published, which is near the Elk Valley in BC, that indicated that they did have quite high mortality in juvenile fish. Are you aware of that paper, I guess, is the question, and have you seen it? And, you know, based on what's in it, would that elevate the level of response from CFIA?
I haven't read the paper, I'll be honest about that. I have heard that there have been some mortalities in the area. It has not been definitively linked to a particular pathogen. And in terms of CFIA's response to that, the Elk Valley is within the Columbia River watershed. So CFIA will not be taking any action against in terms of disease control there. We have limited jurisdiction when it comes to wild animals, even though our whirling disease as a reportable disease does fall under federal jurisdiction. In terms of what is done with wild animals, that a lot of that comes down to either DFO or the province, depending on the area. In this case, the province would have primary responsibility for the wild fish.
Yeah, that Crow's Nest paper, I'll send it your way.
Thank you. Yeah, it's hard to keep up with all of the literature.
Yeah, for sure. Mention infection in the Columbia watershed. What collaboration does CFIA and government have with the Columbia River Salmon Reintroduction Initiative, Indigenous-led collaboration of the Silks Nation, Tanaha, and Squepum Nation?
My engagement, CFIA's engagement with them is somewhat limited. We have reached out to several First Nations to inform them of the as many contacts as we could find to inform them of the detection and of the change. I know that the province of British Columbia is working with some First Nations in the Southern Columbia region to continue the surveillance to determine the spread of the pathogen within the Columbia River watershed, and that is being done in conjunction with several First Nations. We have worked with the Okanagan Nation Alliance in the past on some projects, and we've made them aware of the situation also. However, most of their activities happen within the Columbia River watershed, so our involvement with them is relatively limited.
Okay, there are gonna be a whole bunch of questions around environmental factors and watershed factors. So are you able to give us kind of a synopsis of where we should expect to see high infection rates or higher mortality rates in terms of environmental factors and kind of stream condition as well, if we do end up having whirling disease in these places versus other streams that are maybe colder, less productive, don't have that kind of muddy bottom? Are you able to give us some insight on that?
Well, I'm not a biologist, I will say. So the intimate life cycle information, I'm not completely up on that. But we do tend to find this pathogen in siltier areas. So it needs that tube effects worm to complete its life cycle. So in areas where you do see siltification, then those are areas where you may see a higher prevalence of the pathogen. However, in our surveillance sampling thus far, it's been quite patchy. We've only found, I think in the work we did with Park Canada for the initial detection, we only found partial detections in five pools of fish. So very out of approximately 600 samples. So we're seeing it a very, very low prevalence. And as I said earlier, a lot of those fish didn't show clinical signs. And there were a couple of fish that had suspicious signs of whirling disease that didn't test positive. So there are other pathogens out there that can cause similar clinical signs. So it's really difficult to determine where we may see an increase in this pathogen, the concentration of this pathogen. There is a lot of fish movement, natural fish movement within these waterways in British Columbia. And we're still, we have some information about that, but there's still a lot that's unknown about how and where these fish move. So that's information that we are actively gathering and working with the province to determine. And we're looking at all the options as to how we could try and determine whether there would be areas where we may see clustering of these populations that might have a higher disease prevalence, or if this is more of a widespread situation. And that's information that we'll gather in advance of doing surveillance, but it'll also be informed by the surveillance that we do, the animals that we're able to sample, and the conditions under which we've been able to catch them, about the streams, ponds, lakes where we catch them. That will help to inform us what the type of environment where we may have a greater chance of finding this pathogen, what that environment looks like.
Okay, and then anything around water temperature and the TAMs or how it affects mortality in fish?
As I mentioned earlier, we have not seen high mortality in the populations that we've sampled. So that has been seen in other populations. Certainly, there have been some situations in the US where it was introduced to a naive population and it caused high mortality. It does tend to infect fish through their first summer. So once they swim up and start eating, that's when, as they grow through the season, they become infected by the TAM. We usually target the young of the year fish in the fall, in between August and October, to try and sample these fish and get the best chance of detecting it. So the water temperatures, it is a cold water disease. It doesn't survive freezing. It doesn't survive salinity, but it tends to have that moderate range, which is ideal for salmonids, is basically ideal for the pathogen too.
Okay, does biofilm or benthic cyanobacteria or didymats contribute to the spread of whirling disease? Didymo is able to survive for 40 days outside of water.
It would certainly contribute to the survival of the myxospores. They're quite tough already, so any kind of substrate that they hide in that will insulate them will make it much easier for them to survive. I'm not sure. I don't have any background in the worm habitat and that sort of thing, so I'm not sure whether. We certainly understand that areas with high sediment are more likely to harbor the worm and thus the pathogen. But in terms of the mucus on the fish or any bacterial mats on the bottom of the waterway, I'm sorry, I don't know.
Great. Should marine gear be cleaned as well as if it will be used in fresh water?
No, well, yes. I mean, you should always clean your gear, regardless. But in terms of spread of whirling disease, it's not a concern. The pathogen is not found in salt water. It can't survive any kind of salinity. So we always recommend any gear that's being moved between watersheds, whether it's marine or fresh water, that you apply at least the clean, drain, and dry procedure. But that's not a concern for whirling disease.
Thank you. If a fish is suspected to be infected outside of the Columbia watershed, who should be alerted? Should the fish be kept, and how should the sample be sent in?
You can contact me, or you can email that whirlingdisease@gov.bc.ca email. In terms of what to do with the fish, it can be difficult to say catch the fish, keep it, and then we'll pick it up, because in some cases the storage conditions can affect the ability for us to test. So what I would recommend that you do is take photographs if you can, and take the exact coordinates of the location where you saw the fish, and then let me or the province know about it. And then we can then follow up on that and factor that into our plans. If a fish is seen outside of the Columbia River watershed with concerning signs and it's in the wild, taking that fish out of the environment won't really do much to prevent any spread. So, and it's possible the fish could even survive. So the recommendation is not to capture the fish, but to record it and let us know, and then we can take that into our consideration when we do our surveillance.
Great.
I should say the exception is if you're working with a hatchery, that I want to know about right away, because that's a situation where we may be able to intervene. But with a wild fish, we don't recommend that they be removed from the water.
Thanks. Here's another one. It seems like it would be much more difficult to observe pathogen-associated mortality in a lake compared to a river. How would monitoring for mortality be conducted in a lake?
Well, the fish tend to be quite small when they die. In some cases, you may see them wash up on the shores of the lake, but there are a lot of things that can cause fish kills. It's certainly easier to see in a smaller water body. So in some place like the Arrow Lakes or Shuswap Lake, at those kind of areas that are really, really big, deep lakes, it can be very difficult to monitor for mortalities. But those are also places where when it's very deep, the fish don't like to go that really, really deep into the water when they're that age, because their food source is closer to the shore. So we're more likely to have the right environment for the worms and the pathogen to survive if they're in shallower water or closer to shore in one of these big lakes. So yeah, it's really variable. I mean, it's possible. There's so much going on in these lakes that we really don't understand, that we're really literally scratching the surface to see what we can find. Great.
This one might be a little bit, but it's something people will want to know about, but it might be a little bit outside of your area. There's a clear distinction between a fish infected with M. cerebralis and a fish experiencing whirling disease. There are many examples in the States where the parasite is present, but populations are not being impacted by the disease. And of course, there's the opposite of that too. What is the government of Canada doing to understand the actual risk of whirling disease to our salmonid populations versus presence or absence surveillance of the parasite? I don't know if that's in your shop or if that's outside of your shop.
It's a little outside of my shop. There's a science branch within Canadian Food Inspection Agency, and there are researchers within DFO that are looking at those questions. My expertise is in aquatic animal health and in applying surveillance programs. So there are people doing that work, but it's not me.
Okay, and you might be able to refer us to some of your colleagues, and maybe when we send out an email post webinar.
Yeah, the work is being done, and when studies are completed, they are published. So that information, once the research has been done, it is available in the literature.
Has whirling disease showed up in Waterton Lake? Parks Canada has banned outside watercraft. Is this closing the barn door too late?
We haven't specific. Now the government of Alberta hasn't. If there have been detections in the lake, they haven't let me know, which they're supposed to do. So I think that closure is actually more to do with other invasive species than with whirling disease. It hasn't, as far they're required, laboratories across Canada are required to let CFIA know if they've had a detection, whether it's in an infected area or not. So if they had found it, they would have let me know.
Okay, is it known how long the spore or TAM can live and move without a host?
The TAM doesn't survive for very long. It needs certain conditions. It's quite labile, so it can be in certain conditions. So if the water is too cold or if it gets too warm, then it won't survive. So generally the TAM is in the water column for, I'd have to look at the literature again. I can't remember specifically, but it tends to be there for a few weeks to a month. The myxospores, however, are very tough and can survive for an extended period of time, either in the in a worm or also just in the bottom of the in the substrate at the bottom. So one of the recommendations we have for hatcheries that are dealing with whirling disease is to allow, especially for a silt bottom pond or something like that, is to expose it to extreme temperatures over the winter. And that will kill the myxospores. But other than that, there's, bleach works really well. But if it's just under environmental conditions, it has to be pretty extreme before those myxospores will be destroyed. So yeah, they can last for a long time, years in some cases.
Yeah, that was one of the discussions that we've had with the feds as well. Is like, if you remove the fish, how long does it persist? And I think there's even work going on in Russia and the United States, but the best answer was a long time.
Yeah, we're not entirely certain how long, but it can definitely be several years.
Are there any preservation methods that allow for transport of organs out of the Columbia Basin for research? I saw that freezing was unacceptable. So I think what people are asking with these questions is there a way to preserve a sample to get tested? And they're kind of asking what's the best way.
What sort of samples?
Well, I'm guessing if they suspect that a fish has whirling disease, what should they do if they want to preserve it for testing? What's the best approach?
Well, if you do decide to take the fish out of the water and preserve it for testing, then it should be frozen immediately at -20, preferably in a freezer that doesn't cycle through a defrost. So most fridge freezers, for example, will have an auto defrost, and that can change the temperature of the freezer. So something like a deep freeze is acceptable. No fish should be moved outside of the Columbia River watershed. If you think it may be infected, you should contact CFIA or the province of BC and discuss options. In terms of tissues, some tissue samples for research purposes, that depends on the situation. So if any of you are interested in doing some sampling of salmonid fish, taking other tissue samples, for example, then you should contact me and we can discuss the options.
Awesome. I think that is all that we have for questions. So I really want to thank you, Zoe, for coming on and spending some time with us tonight. Oh, and did I freeze up right at the end? I did. We appreciate you coming on and spending some time with us tonight. That was great. And I think we will take you up on your colleagues too, because there's a lot of interest in there's a lot of questions that are species specific that we probably don't have answers to. And also I think people have a real interest in which systems are potentially higher risk than others as it relates to productivity and water temperature and all those sorts of things. So people are very keen, lots of comments.